Hi everyone, this is Doc Ina, and this is my lecture on Gametogenesis. To download my lecture deck, please go to my WordPress site, Doc Ina Obigaine. These are my main references for this lecture. It's Williams Obstetrics and Comprehensive Gynecology. This is the outline of my lecture. So first, some definition of terms. Oogenesis is the development of the ovum from an oogonium by meiosis. Spermatogenesis, on the other hand, is the development of mature sperm from spermatogonia by meiosis. Conceptional age is the age of the conceptus from fertilization, while gestational age is the stage of the embryo counting from the first day of the last menstrual period. Fertilization is the point at which one spermatozoon penetrates the oocyte. Conceptus is the fertilized oocyte and its derivatives at all stages of development from fertilization until birth and including all extraembryonic membranes. Embryo is the developmental stage of the conceptus after the development of the primitive streak and until all major organs are developed. The zygote is a one-cell stage of the fertilized ovum after pronuclear membrane breakdown but before the first cleavage occurs. Morula is a ball of cells comprising the early embryo that produces both the embryo and the placenta and membranes. Blastocyst is the stage of the conceptus that follows the morula stage. It consists of a fluid-filled cavity surrounded by trophoblasts with embryonic cells at one pole. So when we talk about gametogenesis, we include both the development of the oocytes, which is the oogenesis, and the development of the sperms, which is spermatogenesis. So first, let's talk a little bit about spermatogenesis. Spermatogenesis is a process by which precursor cells termed spermatogonia undergo a complex series of divisions to give rise to spermatozoa. This occurs within the seminiferous epithelium. Proliferation and differentiation of spermatogonia by mitosis occurs in the seminiferous epithelium. Primary spermatocytes, that's 2N or diploid, undergo two meiotic divisions to produce haploid spermatids or N. Approximately 72 days are required to produce mature spermatozoa. Mature spermatozoa, which are haploid, are stored in the caudal epididymis prior to ejaculation. Preservation of optimum sperm function during storage requires adequate testosterone levels in circulation and maintenance of normal scrotal temperature. So here we see a diagram of spermatogenesis. So we have the spermatogonium, which undergoes mitosis to proliferate. So the spermatogonium proliferate by mitotic cell division inside the testis. So, since this is mitosis, we have diploid spermatogonia. So, once it becomes a primary oocyte, it undergoes now the first meiotic division or meiosis 1 to give rise to secondary spermatocytes. The secondary spermatocytes then undergo meiosis 2 or meiotic division 2 to give rise to spermatids, and the spermatids undergo differentiation to become the mature sperm. Let us now discuss ovarian gametogenesis. Oogenesis is the process by which female gametes develop in the ovaries. Unlike in males, all primary oocytes are produced during fetal development. The precursor stem cells in females are called oogonia or oogonium for singular form. Oogenesis begins in fetal life when the primordial germ cells migrate to the germinal ridge by way of the dorsal mesentery of the hindgut by amoeboid action by 6 weeks age of gestation. In the human female, oogenesis begins with the multiplication of the diploid oogonia through multiple rounds of mitosis to produce primary oocytes, reaching about 600,000 by the second month age of gestation and reaching a peak of 6 to 7 million during the first 10 to 12 weeks age of gestation. The numbers then rapidly decline to 2 to 4 million at birth. So by a process of apoptosis and atresia of the enveloping follicle, the number of primary oocytes declines from about 2 to 4 million at birth to become around 90% depleted by puberty. And by age 37, only about 25,000 to 37,000 oocytes remain. And by the age of 50, which is the average age of menopause, only less than 1,000 oocytes remain. Meiosis actually begins at 10 to 12 weeks age of gestation, and it is the mechanism by which diploid organisms reduce their gametes to a haploid state so that they can recombine again during fertilization to become diploid organisms. In humans, this process reduces 46 chromosomes or 23 pairs to 23 chromosome structures in the gamete. The haploid gamete contains only one chromosome for each homologous pair of chromosomes so that it has either the maternal or the paternal chromosome for each pair, but not both. Meiosis is also the mechanism by which genetic exchange is completed through chiasma formation and crossing over between homologous chromosome pairs. In humans, all of this is completed during fetal life of the yet-to-be-born female. The meiotic process is arrested at the diplotin stage, that's the first meiotic arrest, and the oocyte enters the dicutin stages. Primary oocytes remain in the diplotin stage until stimulation by gonadotropins in adulthood during the menstrual cycle. Meiosis I resumes at puberty and is completed at the time of ovulation. The second meiotic division takes place over several hours in the oviduct only after sperm penetration. The primary oocyte is surrounded by a single layer of granulosa cells in a unit referred to as the primordial follicle. Even in the absence of stimulation by gonadotropins, some primordial follicles will develop into primary or pre-antral follicles, at which stage multiple layers of granulosa cells surround them. Development of follicles to this stage appears to be relatively independent of pituitary control, but is probably influenced by intraovarian, nonsteroidal processes that remain to be understood. Development to this stage occurs during the non-ovulatory stages of childhood, pregnancy, oral contraceptive use, as well as during ovulatory cycles. So here's a diagram showing to us folliculogenesis. So from the primordial follicle up to its development to the secondary follicle, that's what we call the pre-antral phase or hormone-independent development. Once it develops an antrum, or it becomes an antral follicle, that is what we call now the antral phase or the hormone-dependent development completion of first meiotic division. With the formation of an antrum, the follicle, which is now the secondary or the antral follicle, enters the final stages of folliculogenesis. So, this is what we call now as the antral phase or the hormone-dependent development completion of the first meiotic division. This is a transition from the intra-ovarian regulation to a major control by the hypothalamic pituitary unit. This requires the presence of the characteristic increase in FSH that occurs in the early menstrual cycle. That is why we call this stage hormone-dependent. This is a diagram of oocyte meiosis, and as we can see here, mitosis and the prophase stages of the first meiotic division occur in the female during fetal life. So we have here an oogonium, which undergoes mitosis in order to proliferate, and then subsequently undergoes meiosis I or first meiotic division. The meiotic process is arrested at the diplotin stage at the 7th month AOG, that's what we call the first meiotic arrest, and the oocyte enters the dicutin stages, that's around the 9th month age of gestation. Meiosis I resumes at puberty and is completed at the time of ovulation, where it undergoes the second meiotic arrest. The second meiotic division takes place over several hours in the oviduct only after sperm penetration, or what we call fertilization. The development process from primary follicle, that's the pre-antral follicle, to secondary or antral follicle, and to a mature pre-ovulatory follicle takes about 1 year to complete. But only about 400 follicles complete this process. The majority of follicles undergo programmed cell death or what we call apoptosis. So in this diagram, we see now that the primordial follicle up to its development into a primary or preantral follicle, that's the gonadotropin independent phase. Once it becomes a secondary or antral follicle, then we have the gonadotropin dependent phase of development. Here now is another diagram showing to us oogenesis and the fertilized ovum. So first, we have the oogonium, which is a diploid, that's a 2N. So during fetal development, all the oogonia differentiate into primary oocytes. And these cells remain frozen in prophase 1 of meiosis, that's the diplotin stage or the first meiotic arrest. When the female reaches puberty, primary oocyte will complete meiosis 1 to produce secondary oocyte, which will remain frozen at the metaphase 2 of meiosis, and that's what we call the second meiotic arrest. Now, during fertilization, the secondary oocyte will complete meiosis 2 to produce the mature ovum, or what we call the egg cell. The follicular phase can be subdivided into 3 periods, namely successive recruitment of a cohort of antral follicles, selection of a dominant follicle, and growth of the selected dominant follicle. For the recruitment of a cohort of antral follicles, FSH provides the critical signal for the recruitment of a cohort of preantral follicles. So this is what we call cyclic recruitment. This is the major survival factor that rescues the follicles from their programmed cell death and allows them to start growing, increasing in size, and beginning to synthesize steroids. Selection of a dominant follicle may be likened to that popular Bible verse that says many are called but only few are chosen. But in this case, for the selection of a dominant follicle, many are called but only one is chosen. Usually only one follicle, that's what we call the dominant follicle, is selected to complete the growth to maturity. Other follicles in the cohort become atretic. So, how was the dominant follicle selected? It's because the dominant follicle is the most well-vascularized or has the most well-vascularized theca layer, allowing therefore a better access of the gonadotropins to their target receptors. This results in a greater local estradiol secretion, which in turn increases the density of the gonadotropin receptors and promotes cell multiplication. So here we can see the dominant follicle that as it increases in size, it increases its secretion of estradiol. And we know that as estradiol increases, it sends a negative feedback to the pituitary to decrease the production of FSH. So if the FSH decreases, the levels of FSH becomes insufficient so that the smaller follicles become atretic and they no longer grow. Production of estradiol requires successive events within different locations in the growing follicle. LH promotes steroid biosynthesis by theca cells and the production of androgens. Now, this androgen, following diffusion into the granulosa layer where the enzyme aromatase is located, are then biotransformed into estradiol. This leads to an overall increase in estradiol production, increased intraovarian estradiol levels, and increased estradiol secretion into the peripheral circulation, which parallels follicular parameter. So the conversion of cholesterol to androstenedione is influenced by LH, and this undergoes in the theca cell. Now, the androstenedione diffuses through the basement membrane to enter the granulosa cell, and this androstenedione will be converted to estradiol under the influence of FSH. This is the reason why this process is also called the two-cell, two-gonadotropin theory. Two cells because that represents theca cell and the granulosa cell, and then the two gonadotropins involved are LH and FSH. As the dominant follicle grows, an antrum or the cavity forms into which follicular fluid accumulates. This fluid inside the cavity contains several steroids, peptide and protein hormones, and nutrients. The dominant follicle increases in both follicular diameter and volume and parallels the increase in the estradiol levels in the blood. At maturation, the dominant follicle reaches a mean diameter range of around 18 to 25 millimeters. Maturation of the dominant follicle is marked by high blood levels of estradiol. When a threshold is reached, estradiol activates the positive feedback loop, and this signals to the hypothalamus and anterior pituitary gland that the follicle is ready for ovulation and that a large gonadotropin surge is to be released. A small but significant increase in the progesterone is also secreted by the follicle before the LH surge. During the ovulatory surge, LH levels increase tenfold over a period of 2 to 3 days, whereas FSH levels increase about fourfold. This gonadotropin surge, or what we call LH surge, is an absolute requirement for the final maturation of the oocyte and the initiation of the follicular rupture. The LH surge initiates germinal vesicle or nucleus disruption, and the fully grown oocyte resumes meiosis, or what we call meiotic maturation. Thus, it progresses from the diplotin stage of the first meiosis to metaphase 2 of the second meiotic division. As the oocyte enters metaphase 2, the first polar body appears. The polar bodies generated from the meiotic events contain relatively little cytoplasm, and the oocyte eventually discards them. At ovulation, meiosis is arrested again. That's what we call the second meiotic arrest. The second meiotic division will only be completed at the time of fertilization. The oocyte's ability to be fertilized coincides with the completion of meiotic maturation. Ovulation or follicular rupture occurs about 32 hours after the initial rise of the LH surge and about 16 hours after the LH surge or the LH peak. Prostaglandins then act locally to induce the hyperemia and edema seen in the first hours of the process of ovulation and which results from increased blood flow and vascular permeability. After the oocyte is extruded from the mature dominant follicles, the amount of follicular fluid is markedly reduced, the follicular wall becomes convoluted, and the follicular diameter and volume greatly decrease. Now, after the oocyte is extruded from the mature dominant follicle, the byproduct will become the corpus luteum. Like the dominant follicle, growth and development of the corpus luteum occur very rapidly. Vascular growth plays a central role in this process. Angiogenic factors such as the vascular endothelial growth factor are present in high quantity in the forming and developing corpus luteum. Progesterone dominance during the luteal phase also affects the hypothalamic thermoregulatory center. Small increase in the basal body temperature reflects increased progesterone secretion during the luteal phase. Thus, the typical BBT curve of the ovulatory menstrual cycle is biphasic. So what happens if fertilization does not occur? The answer to that question, corpus luteum regresses, or this is what we call the process of luteolysis. The lifespan of the corpus luteum is limited to a period of about 14 days, and this is also the reason why the luteal phase of the menstrual cycle is only about 14 days in length. The corpus luteum reaches maturity 8 to 9 days after ovulation, after which time luteal cells start to degenerate and its secretory capability begins to decline, thus becoming the corpus albicans. Rapidly rising concentrations of the human chorionic gonadotropin or the HCG following conception can rescue the corpus luteum and maintain the production of progesterone. The end of the luteal phase is characterized by a dramatic decrease in progesterone, estradiol, and inhibin A, that is the Luteal-Follicular Transition phase. This is accompanied by a characteristic divergence in the FSH-LH ratio, now favoring a specific rise in FSH. The increase in the FSH-LH ratio heralds a new menstrual cycle and the recruitment of a new cohort of follicles. So what happens if fertilization occurs? Now, we see in this picture the journey of the conceptus from the moment the egg cell was fertilized by the sperm up to implantation inside the endometrial cavity. Stage 1 is day 1 to 2, where the sperm meets the egg and the sperm fertilizes the egg and becomes a zygote. Stage 2 is days 2 to 3, where the zygote, which is a one-cell stage, undergoes multiple divisions to become the morula. Stage 3 is days 4 to 5, where the morula develops into the blastocyst and enters the endometrial cavity. And stage 4 is days 5 to 6, when the embryo attaches to the endometrium. The fertilized ovum is what we call the zygote, and this is a diploid. It undergoes slow cleavage for 3 days while still in the fallopian tube. The zygote undergoes multiple cell divisions to eventually become the morula, which is like a solid mulberry-like ball of cells. The morula enters the uterine cavity about 3 days after. Accumulation of fluid between the cells of the morula gives rise to an early blastocyst. Between 3 to 5 days after fertilization, the blastula differentiates into the 58-cell blastocyst. The 58-cell blastocyst composed of 5 embryo-producing cells known as the inner cell mass and the 53 cells destined to be the trophoblast. 6 or 7 days after fertilization, the embryo implants in the uterine wall or the endometrium, and it is composed of 3 steps. First, apposition, which is the initial contact of the blastocyst to the uterine wall. Second is adhesion, where there is increased physical contact between the blastocyst and the uterine epithelium. And lastly, invasion, which is the penetration and invasion of the syncytial trophoblast and cytotrophoblast into the endometrium, which is the inner third of the myometrium, and the uterine vasculature. Successful implantation requires a receptive endometrium appropriately primed with estrogen and progesterone by the corpus luteum. Uterine receptivity is limited to days 20 to 24 of the cycle. If the blastocyst approaches the endometrium after cycle day 24, the potential for adhesion is diminished because anti-adhesive glycoprotein synthesis prevents receptor interactions. Blastocyst loosely adheres to the endometrial epithelium by apposition, and this most commonly occurs in the upper posterior uterine wall. These are some of the terminologies that we use to describe the pregnancy duration. So we have the post-conceptional age, the ovulation age, gestational age, or the menstrual age. For clinical use, we use only gestational age or menstrual age. So how do we compute the gestational age? We use the Naegele rule. So we add 7 days to the first day of the last period and subtract 3 months, then add 1 year. So for example, the last menstrual period or the LMP is July 5, 2016. Then the estimated date of delivery will be July 5 minus 3 months, that will be April 5, plus 7 days, that will be April 12, plus 1 year. So the estimated date of delivery will be April 12, 2017. Human placental formation begins with the trophectoderm, which appears at the morula stage. Trophoblast plays a critical part at the fetal-maternal interface. Its invasiveness promotes implantation, nutritional role for the conceptus, and is essential to maternal physiological adaptations and to pregnancy maintenance. By the 8th day post-fertilization, after initial implantation, the trophoblast has differentiated into an outer multinucleated syncytium, that's the syncytiotropoblast, and an inner layer of primitive mononuclear cells, that's the cytotrophoblast. The syncytiotropoblast is responsible for the synthesis of human chorionic gonadotropin. HCG is responsible for maintaining the corpus luteum. HCG has been detected in the peripheral blood of the mother as early as 6 days after ovulation, but it is always seen by day 12. The concentration doubles every 1.2 to 2 days, reaching its highest point at 7 to 9 weeks of pregnancy. The conceptus is termed an embryo at the beginning of the 3rd week after ovulation and fertilization. Primitive chorionic villi form at this stage. Embryonic period lasts about 8 weeks, during which Oogenesis takes place. The embryonic disc is well defined, and most pregnancy tests that measure human chorionic gonadotropin will become positive by this time. The body stalk is now differentiated during the embryonic period, and the chorionic sac is approximately 1 centimeter in diameter. During the 3rd week, fetal blood vessels in the chorionic villi appear. By the 4th week, a cardiovascular system will have been formed, and thereby a true circulation is established both within the embryo and between the embryo and the chorionic villi. Partitioning of the primitive heart begins in the middle of the 4th week. Arm and leg buds are present and the amnion is beginning to unsheath the body stalk, which therefore becomes or thereafter becomes the umbilical cord. So for my last slide, we discuss the human sex differentiation. Genetic sex is determined at the time of conception. So it will depend whether that embryo has the XX or an XY. So the presence of an Y chromosome is necessary for the development of the testes. Absence of the Y chromosome therefore confers a female phenotype for this conceptus. So primary determinant is the SRY or the testis determining factor gene located on the short arm of the Y chromosome. Testis is responsible for organization with the sexual duct system into a male configuration and suppression of the paramesonephric or Mullerian system. So in the absence of a Y chromosome or in the absence of a gonad, development will be female in nature. That's it. Thank you for watching and please don't forget to subscribe in my WordPress site, Doc Ina Obigayne, and my YouTube, Ina Irabon. Thank you.